So our main focus of tonight's video is going to be on common ancestry and showing the link between organisms and some evidence that will help support that. So our main focus is going to center around the theme of structural evidence to help show common ancestry of eukaryotes. Remember, eukaryotic cells are cells that have membrane-bound organelles. Okay, and so what we're looking for here is we're talking about common ancestry. Okay, so finding that common ancestor, showing that things are conserved and things are similar. And so the first thing we're going to look at here is membrane-bound organelles. So membrane-bound organelles are part of our endosymbiont theory. Okay, if you guys remember from previous biology, right, we talked about endosymbiosis, and we talked about how a larger prokaryote engulfed a smaller prokaryote, and it didn't digest it, it didn't kill it, it ended up being a beneficial relationship for both of them. Okay, and so lipid bubbles are what originally started kind of separating the inside from the outside. Okay, so if you think about it, right, our cell membrane is made of phospholipids, and phospholipids are nonpolar, right? Well, the bulk of them are nonpolar. So they cause a separation, right? They cause a barrier between the fluid. They retain the fluid on the inside, and they keep the fluid on the outside. They make that barrier. So these lipid bubbles began to help separate the insides from the outside. And then over time, we started to develop, you started to see this internal folding, getting these internal membranes that could create small internal microenvironments. And by having those microenvironments, that would allow for the cell to do different functions in different parts. Okay, so my benefit here is that I'm going to get specialization. Okay, I'm going to start to specialize and have certain organelles. I'm going to start to develop organelles that are good for digestion and organelles that are good for making proteins or folding proteins and organelles that make lipids. And so it allows for specialization. And the benefit of specialization within the cell is that's going to increase my cell's efficiency. My cell is going to be a little bit faster, a little bit more competent, able to do a little bit more at one time. Okay, and these cells are the cells that are going to stick around because of natural selection. Right? These cells are, since they are more efficient, okay, they'll become selected for. And so our first big endosymbiotic event was the origin of mitochondria. So what happened was that larger prokaryote, so this larger prokaryote that we can see coming across the bottom here, okay, engulfed that smaller prokaryote that was able to do aerobic respiration, an aerobic bacterium. Okay, but again, it didn't digest it, it didn't break it down. And so that ended up being a beneficial relationship for both of them, right? Remember our mutualism, right? Both of them receive a benefit. And so then those organisms, those cells began to be selected for. Natural selection went into play. And so we started seeing this more and more, it was favored within the population. The second endosymbiotic event was that when the chloroplast was engulfed. So the photosynthetic bacteria this time were engulfed. And again, they were not digested. It was mutually beneficial for both of them. The smaller bacteria got resources so that it could do photosynthesis. It was protected, while the larger cell was getting food. So there's a benefit to both of them. So again, natural selection kicked in, and it favored these types of cells that had engulfed these prokaryotes through this endosymbiosis. Okay, so the presence of chloroplasts and mitochondria do help show evidence of evolution and common ancestry within the eukaryote. Also because the processes that are done in those organelles, okay, the cellular respiration, the photosynthesis that are done there, those processes are basically the same for every organism. And when we see these processes that are conserved like that, okay, we call it a conserved process. So when that process is conserved, basically meaning that we hold on to it, we retain it, we still do it, and the way that it's done doesn't really change. When that process is conserved, that also helps show a link to ancestry. Other structural evidence for common ancestry among eukaryotes is that all eukaryotes, right, so not just some of them, all eukaryotes have linear DNA. mushrooms, birds, trees, they all have linear DNA, and which is different than prokaryotic DNA, right? Prokaryotic DNA is circular. I know this eukaryotic DNA over here looks circular, but you can see there is one end and there is the other. If you pulled it out, it would be an open string, not a closed string like the prokaryotic DNA. So all eukaryotes have linear DNA. So again, this would be an example of a conserved structure. So the way it was built, there wasn't really a better way to build it. So the way it was built has been retained as well as its function. So again, that shows links to common ancestry. In addition, if we look at the genes of the basic gene structure of eukaryotes, that can also show some link to common ancestry. Remember, eukaryotic genes are not continuous. They contain introns. If you think back to protein synthesis, right, remember introns are our junk DNA. So if we look at the picture over on the right here, you can see how the intron is labeled as the orange part. So all of these orange parts here are what are considered junk DNA. So that DNA is not going to be, it's not going to be expressed. Where the exons, the blue, that is the coding DNA. So again, this is different than prokaryotes. Prokaryotic DNA does not have introns. Okay, so again, we're showing this link to common ancestry by having these this similar structure in the way that the DNA is put together. The introns haven't completely been gotten rid of. Okay, and the process of filtering out, pulling out these introns is very similar in all eukaryotic organisms. So the exons are going to be removed by the RNA splicing enzymes. Remember, to splice something means to cut it, to split it. So we have RNA splicing enzymes that are going to come in and they're going to clip those introns. And then what we have next are called SNRPs. I know there's no I in there, but these guys right here, they're pronounced SNRPs. These small nuclear RNA, you can see them, the purple on your picture over here. Okay, they form proteins. Okay, and what they're going to do is they're going to help work with the spliceosome. Okay, they're going to work with the spliceosome and help form the spliceosome. Right, you see our spliceosome here is made of several SNRPs. So now that I've got multiples put together, I now have formed my spliceosome. Remember, again, splice is to cut. So my spliceosome is going to recognize where I want to cut the intron, and it's going to cut it. Okay, it's going to cut the intron. It's going to cut it. It's going to loop it around, and while it's cutting it, it's also going to put the exons together. So now I have the removed intron, but the exons are pasted back together. So the spliceosome will complete all of that. Okay? And so if we again think back to RNA processing, okay, and how that's one of the ways that we can get different products from the same gene, right? The different ways that we splice the exons together. And this is how it's done with these SNRPs and spliceosomes. So one of the things we want to keep in mind as we continue through this evolution unit is that populations of organisms continue to evolve. We didn't just form eukaryotic cells and now we're done. Populations will still continue to evolve. Genomes will change over time, whether that's change in the sequence or change in the size, okay? But they will change. We see that change, right? The fossil record helps show us that change as we see the organisms changing through time, okay? And how new organisms don't just show up randomly, how there is usually a morphological link. We see this happening in organisms that reproduce really quickly. We can see it happen very rapidly, that do generations within weeks or months or maybe years. But again, we see this reproducing very quickly. We see this with our resistance to antibiotics, pesticides, herbicides. Plants reproduce quickly. Bugs reproduce quickly. Bacteria reproduce really fast. Okay? And so when those organisms that are resistant, if they're the ones that are being successful, then they're the ones that are breeding, and they're the ones that are passing their DNA on. And so very quickly, we can see a population of insects or bacteria, we can see them change. We can see that shift. We could do the calculations and we can see the allele frequencies shift. Okay? And one of the issues with that is pathogenic bacteria and viruses, those can start to cause disease, right? Where the bacteria or virus may not have been harmful at first because of how rapidly it changes, then they may not respond to medicines that used to work for them. They may not respond, to our immune system quickly. We see that with Zika and SARS are both examples of that. If you look at this picture over here on the right, there's Zika there circled in red. Okay, and there's just a 20% nucleotide sequence divergence between those surrounding viruses. So just not a huge change in the nucleotide sequence, but it changes very rapidly, okay? And we get all of these different viruses, these different diseases that seem to pop up out of nowhere. Okay? So again, just something we need to keep in mind as we talk about this, that this is an ongoing, continuous process that has never stopped. So like always, we'll continue to look at data and we will continue to practice writing about this.